So let's talk about biology, and I mean really talk about it. The whole story, from the very beginning all the way to where we are right now. Because here's the thing, most of us sat through biology class memorizing the parts of a cell, labeling diagrams of the human heart, maybe dissecting a frog if we were lucky, or unlucky, depending on how you felt about that. But nobody ever told us the actual story behind all of it. And that story is wild. We're talking thousands of years of humans trying to figure out one simple question. What is life? How does it work? And why does it do what it does? That question has led to plagues that wiped out entire civilizations. A quiet monk spending 8 years in a pea garden unlocking the secret of heredity. A 5-year voyage on a ship called the Beagle that shook the foundations of everything people believed. 2 scientists in a desperate race to decode the very blueprint of life itself. And right now, today, a technology that lets us edit living organisms like you'd edit a document on your laptop. Before we get into all of that though, I need to set something straight. When you think of biology today, you probably picture microscopes and DNA helixes and maybe some complicated diagrams of how cells divide. But biology hasn't always looked like that. For most of human history, biology was just people looking at the world around them, watching animals, picking apart plants, cutting open bodies, and trying to make sense of what they saw. No labs, no technology, just observation and a lot of guessing. And somehow, from those raw beginnings, we built everything we know today. So where do we even start? Well, let me take you back. Way back. We're going back to the ancient world. And I'm talking ancient, because humans have been trying to understand living things for as long as humans have existed. But the first people to really start writing it down, organizing it, thinking about it systematically, that takes us back thousands of years across multiple civilizations. Let's start with Egypt. Now, the ancient Egyptians are famous for pyramids and pharaohs, but they were also some of the earliest people to seriously study the human body. And they had a pretty good reason for it. Mummification. When you're preserving dead bodies as a religious practice, you end up learning a lot about what's inside them. Egyptian embalmers knew about the major organs. They knew the heart, the lungs, the liver, the intestines. They carefully removed and preserved these organs in canopic jars. But here's what's interesting. They threw away the brain, just scooped it out through the nose and tossed it. They thought the heart was the center of intelligence and emotion, not the brain. The brain was just stuffing as far as they were concerned. They were wrong about that, obviously, but their medical knowledge was actually pretty impressive for the time. There's this document called the Edwin Smith Papyrus, written around 1600 BCE, and it's basically an ancient surgical textbook. It describes 48 different injuries and how to treat them. Head wounds, broken bones, dislocations. And the remarkable thing is how rational it is. No magic spells, no prayers to the gods, just careful observation. If you see this injury, do this treatment. If the patient shows these symptoms, the prognosis is this. It's shockingly modern in its approach. Then there's the Ebers Papyrus, which is more of a medical encyclopedia. It lists hundreds of remedies using plants, minerals, and animal parts. Some of it is nonsense by our standards, but some of it actually works. They used honey as an antibacterial agent on wounds. Modern science has confirmed that honey genuinely does have antibacterial properties. They were onto something without fully understanding why. Now, let's jump over to the Indian subcontinent, because what was happening there is genuinely remarkable. In ancient India, there was a medical tradition called Ayurveda, and it goes back thousands of years. The 2 big texts are the Karaka Samhita and the Sushruta Samhita. Karaka focused on internal medicine. He classified diseases, described their causes, and laid out treatments. He talked about digestion, metabolism, immunity. He even discussed the idea that tiny invisible living things could cause disease. This is thousands of years before germ theory became accepted in Europe. Let that sink in for a moment. But Sushruta is the one who really blows your mind. This guy was a surgeon, and not just any surgeon. He's often called the father of surgery. The Sushruta Samhita describes over 300 surgical procedures and 120 surgical instruments. He wrote about how to perform cataract surgery, how to fix broken bones, how to do skin grafts. In ancient India, he developed a technique for reconstructing noses using a flap of skin from the forehead. This technique, with modifications, is still used today. It's called the Indian flap method. And Sushruta emphasized something that modern medicine considers absolutely essential. He said that a surgeon should practice on dead animals and on fruits and vegetables before operating on living patients. That's basically the same principle behind modern surgical training. Practice before you cut into a real person. Over in China, there was a completely different but equally fascinating tradition. Chinese medicine goes back thousands of years, and one of the foundational texts is the Shennong Bencao Jing, attributed to the legendary Emperor Shennong, who supposedly tasted hundreds of herbs to test their properties. The story goes that he poisoned himself dozens of times in the process. Whether that's true or not, the text catalogs hundreds of medicinal plants, classifying them into categories based on their effects. Chinese scholars were meticulous about observing nature. They developed detailed systems for classifying plants and animals. They had sophisticated ideas about how the body works, built around concepts like qi and the balance of yin and yang. Different framework than what we use today, sure, but the underlying impulse was the same. Watch carefully, find patterns, try to understand how living systems work. But if we're talking about where biology as a real discipline of inquiry began, we have to talk about ancient Greece, because the Greeks did something different from everyone else. They didn't just observe and catalog. They asked why. They wanted explanations. They wanted theories. And the biggest name here is Aristotle. This guy was something else. Aristotle, who lived in the 4th century BCE, is legitimately the first real biologist in history. He didn't just study one type of living thing. He studied everything: animals, plants, marine life. He personally dissected animals to understand their anatomy. He observed chicken embryos developing inside eggs, day by day, documenting the changes. He classified around 500 different animal species, organizing them by their characteristics. He distinguished between animals with blood and animals without blood, which roughly maps onto our modern division between vertebrates and invertebrates. He noticed that dolphins breathe air and nurse their young, so he classified them as mammals, not fish. This was correct, and it took European science another 2,000 years to catch up to that observation. Aristotle's student Theophrastus picked up where his teacher left off, but with plants. Theophrastus is called the father of botany. He described and classified hundreds of plant species. He wrote about how plants grow, how they reproduce, how soil and climate affect them. His works were the most important botanical texts in the world for nearly 2,000 years after his death. Then there's Hippocrates, who changed medicine forever with one radical idea: disease is natural, not supernatural. Before Hippocrates, most people thought illness was punishment from the gods or the result of evil spirits. Hippocrates said no. Diseases have natural causes, and they can be understood and treated through observation and reason. He developed the theory of the 4 humors: blood, phlegm, yellow bile, and black bile. He said health was about keeping these 4 substances in balance. If they got out of balance, you got sick. Was the theory correct? No, not really, but the approach was revolutionary. Look at the patient, observe the symptoms, think about what's causing them. Try treatments and see what works. That's the foundation of clinical medicine. And then there's Galen. Galen lived in the 2nd century CE in the Roman Empire, and he became the most influential medical authority in history. He was the personal physician to Roman emperors. He got a lot of his anatomical knowledge from treating gladiators, patching up their wounds, seeing the insides of living humans in ways that most doctors never did. He also dissected animals extensively, especially monkeys and pigs, trying to understand human anatomy by comparison. Galen wrote extensively about the body's systems. He described muscles, nerves, the circulatory system, the digestive system, and he was brilliant. But here's the problem. He also got a lot of things wrong. He thought the liver was the center of the circulatory system, that blood was continuously produced and consumed rather than circulating. He made errors in anatomy because he was often dissecting animals and assuming humans were the same. And because of his enormous authority and reputation, nobody questioned him. For 1,400 years, if Galen said it, it was true. Doctors in medieval Europe and the Islamic world treated his texts like scripture. If what they saw in a dissection didn't match what Galen wrote, they assumed the body was wrong, not Galen. That's how powerful his authority was. And this is the tension that defined early biology: observation versus authority, curiosity versus tradition. The Greeks were brilliant at asking questions and making observations, but they also had a tendency to let their theories and philosophies override what their eyes were telling them. And once someone like Galen became the established authority, it took over 1,000 years before anyone dared to seriously challenge him. But challenge him they did, and that's where the story gets really interesting. Show more, Feifinger PM. Now here's something that most Western textbooks completely skip over, and it drives me crazy because it's one of the most important chapters in the entire history of biology. After the fall of Rome, Europe went into a long intellectual slump. The great Greek and Roman texts were gathering dust. Libraries were crumbling. Scientific inquiry basically ground to a halt. But on the other side of the Mediterranean, something incredible was happening. The Islamic world was entering its golden age, and biology, medicine, and the study of living things were about to take a massive leap forward. It started with translation. Under the Abbasid Caliphate, centered in Baghdad, there was this extraordinary institution called the House of Wisdom. Scholars from all backgrounds, Muslim, Christian, Jewish, Persian, were brought together with one mission: translate everything, every Greek text they could find. Aristotle, Galen, Hippocrates, Theophrastus, all of it was translated into Arabic, studied, debated, and then expanded upon. This wasn't just preservation, this was active scholarship. These scholars didn't just copy the Greek texts and call it a day. They read them, tested them, found the mistakes, and pushed the knowledge further. Without this translation movement, most of the Greek scientific works would have been lost forever. Let that sit for a second. A huge chunk of what we consider the foundation of Western science survived only because Arabic scholars thought it was worth saving and improving. One of the most fascinating figures from this era is Al-Jahiz. This guy lived in the 9th century in Basra, and he wrote this massive work called Kitab al-Hayawan, the Book of Animals. It's 7 volumes, and what's remarkable about it is that Al-Jahiz wasn't just listing animals and describing what they look like. He was thinking about how they relate to each other and to their environments. He described food chains. He talked about how animals struggle to survive, how they compete for resources, how the environment shapes which animals thrive and which ones don't. He observed that animals in different regions develop different characteristics suited to their surroundings. This is proto-evolutionary thinking. Nearly 1,000 years before Darwin, Al-Jahiz was writing about the influence of environment on organisms and the struggle for existence. He didn't have the full theory of natural selection, obviously, but the seeds of those ideas are right there in his work, and almost nobody in the West knows about it. Then there's Ibn Sina. In the West, they call him Avicenna, and this man wrote what might be the single most influential medical textbook in human history. It's called Al-Qanun fi al-Tibb, the Canon of Medicine. He finished it around 1025 CE, and it's a monster of a book. 5 volumes covering everything: general medical principles, simple drugs, diseases organized by organ system, diseases that affect the whole body, and compound drugs. Ibn Sina synthesized Greek, Indian and Islamic medical knowledge into one coherent system. He described the contagious nature of certain diseases. He understood that tuberculosis was infectious. He discussed the connection between emotional health and physical health. He laid out principles for testing the effectiveness of drugs that sound remarkably like modern clinical trial criteria. You need to test a drug in its pure form on a simple disease first, on multiple patients, and observe whether the effect is consistent. That is scientific methodology from the 11th century. The canon of medicine was used as the primary medical textbook in both the Islamic world and European universities for over 600 years, 6 centuries. Nothing else in the history of medical education even comes close to that kind of longevity. Now here's a story that should make you genuinely angry about how history is taught. Ibn al-Nafis, a 13th century physician working in Damascus and Cairo, correctly described pulmonary circulation. He explained how blood flows from the right side of the heart to the lungs, gets mixed with air, and then flows to the left side of the heart. He directly contradicted Galen, who had said that blood passes through invisible pores in the wall between the two sides of the heart. Ibn al-Nafis said no, there are no pores. The blood must pass through the lungs. He was absolutely right. And this was 300 years before William Harvey got the credit for describing circulation in Europe. 300 years. Ibn al-Nafis figured it out first, wrote it down, and the world mostly forgot about it until his manuscripts were rediscovered in the 20th century. And we can't skip al-Dinawari. He's called the founder of Arabic botany, and for good reason. His book of plants classified and described over 600 plant species. He documented their growth stages from germination to death, described how different soils and water conditions affected plant development, and organized his observations systematically. This was serious botanical science, not just a list of herbs and what they cure. Now, while all of this brilliance was happening in the Islamic world, what was going on in Europe? Honestly, not much. Medieval Europe was intellectually stagnant when it came to biology and medicine. Physicians relied almost entirely on Galen and Aristotle. The idea of questioning these ancient authorities was borderline heretical. Dissecting human bodies was heavily restricted. Medical training was based more on reading old texts than on actually examining patients. There were some bright spots, though. Monastic communities maintained herb gardens and compiled herbals, books describing medicinal plants. Hildegard of Bingen in the 12th century wrote about natural history and medicine, but these were exceptions, not the rule. For the most part, European biology was frozen in place, repeating what the Greeks and Romans had said over 1,000 years earlier, while the Islamic world was actively advancing the science. But things were about to change. As European Christians conquered Islamic territories in Spain and Sicily during the Reconquista, they gained access to those incredible Arabic libraries. Scholars started translating Arabic texts into Latin, and suddenly, Europeans had access to not just the original Greek works, but all the advancements Islamic scholars had built on top of them. This influx of knowledge, combined with a growing restlessness, a desire to look at the world with fresh eyes, set the stage for what came next, the Renaissance. And in the Renaissance, people finally started doing something radical. They stopped just reading about the human body. They started cutting it open and looking for themselves. The Renaissance changed everything. And I mean everything. Because after centuries of just reading old books and trusting ancient authorities, people in Europe finally started doing something that sounds so simple, but was absolutely revolutionary at the time. They started looking for themselves. And nobody embodies this shift more than Andreas Vesalius. This guy was a Belgian anatomist, born in 1514, and he did something that sounds obvious, but was genuinely radical in the 16th century. He actually dissected human bodies and carefully documented what he found. Now, dissection wasn't entirely new, but here's the difference. Before Vesalius, when professors taught anatomy, they'd sit up in a high chair reading from Galen's texts while an assistant did the actual cutting. The professor never even looked at the body. Whatever Galen said was in there, that's what was in there. End of discussion. Vesalius said forget that. He got down from the chair, picked up the scalpel himself, and started cutting. And what he found was shocking. Galen was wrong. A lot. Galen had been dissecting monkeys and pigs and assuming humans were the same. Vesalius, actually looking at human bodies, found over 200 errors in Galen's descriptions. The jawbone isn't 2 pieces, it's 1. The liver doesn't have 5 lobes, it has a different structure. The sternum isn't made of 7 segments. On and on and on. 200 plus corrections to the man whose word had been law for 1,400 years. In 1543, Vesalius published De Humani Corporis Fabrica, on the fabric of the human body. It's a stunning work. 7 volumes with incredibly detailed anatomical illustrations that were revolutionary for their accuracy. The drawings show muscles, bones, organs, and the nervous system with a precision nobody had achieved before. And the message of the book was clear. Don't trust authority. Trust your eyes. Look at the actual body. Observe what's really there instead of what someone told you should be there. And here's a beautiful coincidence. 1543 is the same year Copernicus published his book, showing that the Earth orbits the Sun, not the other way around. In the same year, 2 men fundamentally challenged the old authorities. Copernicus challenged Ptolemy's model of the universe. Vesalius challenged Galen's model of the human body. 1543 might just be the most important year in the history of science, but Vesalius was just the beginning. About a century later, an English physician named William Harvey took this new spirit of actually looking and testing and pushed it even further. Harvey wanted to understand the heart and blood. Galen had taught that blood was produced by the liver, flowed outward through the body, and was consumed by the organs. Blood didn't circulate. It was constantly being made and used up. This had been accepted for over 1,000 years. Harvey said, "Let me actually test this." He did experiments. He measured how much blood the heart pumps with each beat. He calculated the volume. And he realized that if Galen was right, the liver would have to produce an absurd amount of blood every single hour, way more than the body could possibly manufacture. The math didn't work. So Harvey proposed something radical. Blood circulates. The heart is a pump. It pushes blood out through arteries. The blood travels through the body and then returns through veins back to the heart. A closed loop. Continuous circulation. He published this in 1628 in a work called De Motu Cordis on the motion of the heart, and people resisted it. Of course they did. Harvey was overturning 1,400 years of established medical teaching. Some doctors attacked him viciously. His medical practice actually suffered because people thought he was a crackpot. But he had the evidence, he had the experiments, he had the math, and eventually, the truth won out. Harvey proved something incredibly important, not just about blood, but about science itself. Experimentation beats authority. It doesn't matter how famous or ancient the source is. If the evidence says otherwise, the evidence wins. Now here's where the story takes a turn that nobody saw coming. Because up to this point, everything biologists studied was visible to the naked eye. Animals, plants, organs, bones. You could see it all if you just looked carefully enough. But what if there was an entire world of living things that you couldn't see? What if life existed at a scale so small that no human eye could detect it? In the 1660s, a new invention blew that door wide open: the microscope. Robert Hooke was an English scientist and one of the most curious minds of his era. In 1665, he published a book called Micrographia, and it became an instant sensation. Hooke had used a microscope to look at all sorts of things: the surface of a razor blade, the eye of a fly, a thin slice of cork. And when he looked at that cork under magnification, he saw something strange. It was made up of tiny rectangular chambers, like little rooms. He called them cells because they reminded him of the small rooms monks lived in. That word stuck. And it turns out Hooke had stumbled onto something massive. He didn't fully realize it at the time, but he was seeing the basic building blocks of all life. Every living thing on this planet is made of cells. Hooke saw them first, but it was a Dutch tradesman named Antony van Leeuwenhoek who really blew the lid off. Leeuwenhoek wasn't a trained scientist. He was a fabric merchant who made his own microscopes as a hobby, but his microscopes were better than anyone else's, way better. And when he started looking at things under his lenses, he discovered an invisible universe. He looked at pond water and saw tiny creatures swimming around. He called them animalcules. We now know these were bacteria, protists, and other microorganisms. He looked at dental scrapings and saw bacteria. He examined blood and saw red blood cells. He observed sperm cells for the first time in history. Leeuwenhoek wrote letters to the Royal Society in London describing what he saw, and people could barely believe it. An entire hidden world of living things, too small to see with the naked eye, teeming everywhere, in water, in your mouth, in your blood. This was a revelation. Life wasn't just the animals and plants you could see around you. Life existed at scales nobody had ever imagined. And if there were living things that small, what else didn't we know? Meanwhile, something else was driving biology forward: European exploration. Ships were sailing to every corner of the globe and coming back with plants and animals that nobody in Europe had ever seen before. Thousands of new species from the Americas, Africa, Asia, the Pacific, exotic birds, strange fish, unfamiliar flowers, bizarre insects. Natural historians were overwhelmed. Collections were growing out of control, and a really practical problem was emerging. How do you organize all of this? How do you name it? How do you make sense of tens of thousands of different living things? There was no consistent system. Different scholars used different names for the same species. Descriptions were long and confusing. It was chaos, and that chaos was demanding a solution. Someone needed to bring order to the living world. Someone needed to build a system for naming and classifying every living thing on Earth. And someone did. So picture the situation. It's the 1700s. Ships are arriving in European ports from every corner of the planet, carrying crates full of dried plants, preserved animals, seeds, shells, insects pinned to boards. Natural history collections are exploding in size, and every naturalist has their own system for naming things. One guy calls a plant by a long Latin description that's basically a full sentence. Another guy calls the same plant something completely different. A third guy doesn't even know it's the same plant because the first two descriptions sound nothing alike. It was a mess, a genuine scientific mess. You couldn't share discoveries if you couldn't even agree on what to call things. Biology desperately needed someone to bring order to the chaos. Enter Carl Linnaeus. This Swedish botanist, born in 1707, was borderline obsessed with classification, and I mean obsessed. From a young age, he was cataloging plants, organizing them, sorting them into groups. He believed that God had created all living things in a perfect order, and it was humanity's job to discover and document that order. And in 1735, at just 28 years old, he published the first edition of Systema Naturae. It was only about a dozen pages at first, but by the time Linnaeus was done revising and expanding it over the following decades, it had grown into a massive comprehensive catalog of the natural world. Here's what made Linnaeus revolutionary. He introduced binomial nomenclature, two-name naming. Every species gets exactly two Latin names. The first is the genus, the group it belongs to. The second is the specific name, identifying that particular species within the genus. So humans aren't described by some long rambling Latin phrase anymore. We're Homo sapiens. The domestic cat is Felis catus. The common sunflower is Helianthus annus. 2 words: clean, universal. A scientist in Sweden and a scientist in Japan could now talk about exactly the same organism without any confusion. This sounds so simple that it's easy to underestimate how transformative it was. Before Linnaeus, there was no standard. After Linnaeus, there was a universal language for biology, and we still use it today. Every species discovered right now in the 21st century gets a binomial Latin name following Linnaeus's system. That's nearly 300 years of continuous use. It's one of the most successful inventions in the history of science. But Linnaeus didn't just name things, he organized them into a hierarchy. He grouped species into genera, genera into orders, orders into classes, classes into kingdoms. The system has been refined since then. We've added phylum and family and adjusted the categories. But the basic idea of a nested hierarchy, smaller groups sitting inside bigger groups, that's pure Linnaeus. Kingdom, phylum, class, order, family, genus, species. If you ever memorized a mnemonic for that in school, you have Linnaeus to thank, or blame, depending on how that exam went. Now here's the thing about Linnaeus that's important for where this story goes next. Linnaeus believed that species were fixed, permanent, unchanging. God created every species exactly as it is, and that's how it would stay forever. New species don't appear. Old species don't change. The job of the naturalist is just to catalog what God already made. This was the dominant view at the time. And Linnaeus's classification system, in a way, reinforced it. Everything has its place. Everything fits neatly into its box. The natural world is orderly and static, but not everyone agreed. And one of the most important dissenters was a French naturalist named Georges Louis Leclerc, Comte de Buffon. Buffon was a contemporary of Linnaeus, and the two did not get along. Buffon thought Linnaeus's obsession with rigid classification was actually misleading. Nature isn't that neat, Buffon argued. Nature is messy, variable, and constantly in flux. Buffon wrote this enormous work called Histoire naturelle, 36 volumes published over his lifetime and another 8 after his death. It covered everything in the natural world. And buried within those volumes were some genuinely radical ideas. Buffon suggested that species might change over time, that the environment could shape organisms, that animals in different parts of the world might have diverged from common ancestors. He pointed out the similarities between humans and apes and suggested they might share a common origin. He proposed that the Earth was far older than the biblical timeline suggested, maybe 75,000 years old or more. He was actually way off on that number. The Earth is about 4.5 billion years old. But the point is that Buffon was pushing against the idea that everything was fixed and static. He was saying the living world might have a history. It might have changed. It might still be changing. Buffon didn't have a mechanism to explain how species change. He didn't have a full theory, but he was planting seeds, dangerous seeds. Because once you start classifying organisms and putting them into groups based on their similarities, a really uncomfortable question starts to emerge. Why are some species so similar to each other? Why do humans and apes share so many features? Why do different species of finches look almost identical except for their beaks? Why do fossils show creatures that look like modern animals but aren't quite the same? If every species was created separately and perfectly by God, why does the living world look like it has a family tree? That question was going to haunt biology for the next century, and the answer, when it finally came, would shake the foundations of not just science, but religion, philosophy, and how human beings understand their place in the universe. Before Darwin, before anyone uttered the word evolution in a scientific context, the ground had to be prepared, and that preparation came from an unexpected place: rocks. See, for most of human history, people assumed the Earth was young. Biblical scholars had calculated that God created the world around 4000 BCE, making the planet roughly 6,000 years old. And if the Earth was that young, there wasn't enough time for anything to change. Species were created as they are. The world was static. End of story. But in the late 1700s, a Scottish farmer and geologist named James Hutton started looking at rock formations and seeing something that didn't fit. He saw layers upon layers of sediment built up over what had to be incredibly long periods of time. He saw rocks that had been pushed up, worn down, covered by new layers, and pushed up again. Cycles within cycles that required not thousands, but millions of years. Hutton proposed that the Earth was ancient, unimaginably old, and the same geological processes we see today, erosion, volcanic activity, sedimentation, these had been operating slowly and steadily for all that time. He called this uniformitarianism. The present is the key to the past. A few decades later, Charles Lyell picked up Hutton's ideas and ran with them. Lyell published Principles of Geology starting in 1830, and it became hugely influential. He laid out detailed evidence that the Earth was millions upon millions of years old. And here's why this matters for biology. If the Earth is ancient, then there's been enough time for living things to change. A young Earth means a static world. An old Earth opens the door to transformation. Lyell's book, by the way, was one of the books a young Charles Darwin took with him on a certain sea voyage, but we'll get to that. Meanwhile, fossils were causing problems for anyone who believed the natural world was unchanging. People had been finding strange bones and shells embedded in rocks for centuries, but the standard explanation was that these were either tricks of nature, remains from the biblical flood, or just oddities. Then along came Georges Cuvier. Cuvier was a French naturalist working in the early 1800s, and he was brilliant at comparative anatomy. He could look at a handful of bones and reconstruct what the entire animal looked like, and what he found was devastating to the old world view. He demonstrated beyond any doubt that some of these fossils belonged to species that no longer exist anywhere on Earth. They were gone, extinct, forever. This was a shocking idea. If God created every species perfectly, why would any of them disappear? Cuvier himself didn't believe in evolution. He thought catastrophic events, floods, earthquakes, periodically wiped out species, and then new ones appeared through separate creation events. But the fundamental observation stood. Species go extinct. The living world is not permanent. Things that once existed can vanish, and things that exist now weren't always here. The ground was shifting under everyone's feet. Now, the first person to put together a real comprehensive theory of how species change over time was Jean-Baptiste Lamarck. Lamarck was a French naturalist, and in 1809 he proposed something bold. Species aren't fixed. They transform over generations. They adapt to their environments and pass those adaptations to their offspring. His most famous example is the giraffe. A giraffe stretches its neck to reach high leaves. That stretching somehow makes its neck slightly longer. Its offspring inherit the longer neck. Over many generations, you get the long-necked giraffes we see today. This is called the inheritance of acquired characteristics, and it's wrong. At least the mechanism is wrong. Stretching your neck doesn't change the DNA you pass to your kids. A bodybuilder's children aren't born with bigger muscles. It doesn't work that way. But here's what people often miss about Lamarck. His instinct was right. Species do change over time. They do adapt to their environments. Those adaptations are inherited by future generations. Lamarck had the right big picture, but the wrong mechanism, and he was viciously mocked for it. Cuvier in particular attacked Lamarck's ideas and basically destroyed his reputation. Lamarck died in 1829, blind, poor, and largely forgotten. His daughter reportedly said at his funeral that posterity would admire him. She was eventually proven right, but it took a long time. So by the early 1800s, all the puzzle pieces were on the table. The Earth is ancient. Species go extinct. Fossils show creatures that look similar to, but different from modern species. Living things seem to be adapted to their environments in suspiciously specific ways. Everything was pointing toward the idea that life changes over time, but nobody had figured out how. Nobody had the mechanism. And then a 22-year-old English naturalist stepped onto a ship called the Beagle. Charles Darwin boarded HMS Beagle in 1831 for what was supposed to be a 2-year survey voyage. It turned into 5. And those 5 years changed the course of human history. Darwin visited South America, the Galapagos Islands, Australia, and dozens of other locations. He collected specimens obsessively: fossils, birds, insects, plants, anything he could get his hands on. and he observed. He noticed that the finches on different Galapagos islands had different shaped beaks. Some had thick beaks for cracking seeds, others had thin beaks for catching insects. They were clearly related, clearly similar, but each one was perfectly suited to the specific food sources on its particular island. Why would God create separate but almost identical species for each tiny island? It didn't make sense unless these birds had once been the same species and had gradually changed over time to fit their different environments. But Darwin didn't rush to publish. And this is one of the most fascinating parts of the whole story. Darwin figured out the basic idea of evolution by natural selection within a few years of returning from the voyage. He wrote it down in private notebooks. He knew what he had, and he sat on it for 20 years. 20 years of silence. Why? Because Darwin understood exactly how explosive this idea was. It didn't just challenge scientific orthodoxy, it challenged religion. It challenged the idea that humans were special creations made in God's image. If species evolve through natural processes, then humans are just another animal, shaped by the same forces as every beetle and barnacle. Darwin was terrified of the backlash. He was a quiet, private man from a respectable family. His wife Emma was deeply religious. He knew this theory would cause an uproar, so he kept collecting evidence, mountains of it. He spent 8 years just studying barnacles, 8 years on barnacles. He wanted his case to be so overwhelming that nobody could dismiss it. And then in 1858, a letter arrived that changed everything. Alfred Russel Wallace, a young naturalist working in Southeast Asia, had independently come up with the same theory: natural selection, survival of the fittest. Wallace wrote to Darwin asking for his opinion, and Darwin was stunned. Someone else had figured it out. After 20 years of careful secrecy, he was about to get scooped. Darwin's friends arranged a compromise. Both Darwin's and Wallace's papers were presented together at the Linnean Society in London in 1858. And then Darwin finally got to work finishing his book. On the Origin of Species was published on November 24th, 1859. The first printing sold out on the first day. The theory is elegant in its simplicity. Within any population, individuals vary. Some are slightly taller, some faster, some better camouflaged. These variations are heritable, passed from parent to offspring. Resources are limited. Not everyone survives. The individuals whose variations give them an advantage in their particular environment are more likely to survive and reproduce. Their advantageous traits get passed to the next generation. Over vast stretches of time, these small advantages accumulate. Populations change. New species emerge. No divine intervention required. Just variation, inheritance, selection, and time. The response was immediate and fierce. The church was outraged. Many scientists were skeptical. The most famous confrontation happened in 1860 at Oxford, where Thomas Henry Huxley, Darwin's most aggressive defender, debated Bishop Samuel Wilberforce. The bishop sarcastically asked whether Huxley claimed descent from an ape on his grandfather's side or his grandmother's side. Huxley reportedly responded that he would rather be descended from an ape than from a man who used his gifts to obscure scientific truth. The crowd erupted. It became one of the most legendary moments in scientific history. Huxley earned the nickname Darwin's Bulldog for his fierce public defense of evolution, because Darwin himself hated confrontation and avoided public debates entirely. But here's what's important. Darwin won, not immediately, not completely, but within a generation, the scientific community largely accepted that evolution happens. The evidence was just too strong. Fossils, biogeography, comparative anatomy, embryology, everything pointed in the same direction. Life evolves, species change, humans share ancestors with other primates. The tree of life is real, and yet Darwin had a massive problem. A hole in his theory so big that it kept him up at night. He could explain that traits vary and that advantageous traits get selected, but he could not explain how. How do traits get passed from parent to offspring? what is the actual mechanism of inheritance? Darwin proposed a theory he called pangenesis, where tiny particles from all over the body collect in the reproductive organs and get passed to offspring. It was completely wrong. He knew it was weak. His critics knew it was weak. Without a mechanism for heredity, the whole theory had a gaping hole in its foundation. And here's the part that's almost too perfect to be true. While Darwin was agonizing over this problem, while scientists were debating whether evolution could work without understanding heredity, the answer already existed. It had already been discovered. In the 1860s, right around the time Origin of Species was shaking the world, a quiet Augustinian monk in a monastery in Brno was carefully crossbreeding pea plants in his garden. He was tracking how traits like height, flower color, and seed shape passed from one generation to the next. He was finding precise mathematical ratios. He was uncovering the fundamental laws of heredity, everything Darwin needed. The monk published his findings in 1866 in an obscure local journal. Almost nobody read it. Darwin never saw it. The scientific world completely ignored it. The monk died in 1884 thinking his work had been a failure. His name was Gregor Mendel, and his paper sat unread in library shelves for 34 years before anyone realized he had already solved the puzzle that Darwin couldn't. His name was Gregor Mendel, and his story is one of the most heartbreaking in all of science, because this quiet, unassuming monk figured out the laws of heredity decades before anyone else, and nobody cared. Mendel was born in 1822 in what's now the Czech Republic. He came from a poor farming family and entered the Augustinian monastery in Brno, partly because it gave him access to education he couldn't otherwise afford. The monastery was actually a center of learning, and Mendel studied physics, mathematics, and natural history. He even went to the University of Vienna for 2 years, and then he came back to the monastery and started growing peas, not casually, obsessively. Between 1856 and 1863, Mendel cultivated nearly 29,000 pea plants. 29,000. He carefully controlled which plants bred with which. He tracked specific traits across generations: plant height, tall or short, seed shape, round or wrinkled, seed color, yellow or green, flower color, purple or white. 7 traits in total, and he counted everything. This is what made Mendel different from every naturalist before him. He didn't just observe, he quantified. He turned biology into mathematics, and what he found was stunning. When he crossed a tall plant with a short plant, the first generation was all tall. The short trait seemed to disappear, but when he crossed those tall offspring with each other, the short trait came back in the next generation, and not randomly, in a precise ratio, 3 tall to 1 short. Every time, across thousands of plants, 3 to 1, Mendel realized that each trait is controlled by what he called factors. We now call them genes. Each organism inherits 2 factors for each trait, 1 from each parent. Some factors are dominant. They show up even if you only have 1 copy. Others are recessive. They only show up if you have 2 copies. That first generation was all tall because tall is dominant over short. But each tall plant was secretly carrying 1 copy of the short factor. When 2 of those carriers bred, 1/4 of their offspring got 2 short factors and ended up short, 3 to 1. This was the mechanism Darwin was missing. Traits don't blend together like mixing paint. They're carried as discrete units that get shuffled and dealt like cards in every generation. This explained how variation is maintained in a population. This explained how a trait could skip a generation and reappear. This was the foundation of everything we now call genetics. Mendel published his results in 1866 in the Proceedings of the Natural History Society of Brno. It was a local journal with limited circulation. He sent copies to about 40 other scientists and institutions. Almost nobody responded. The few who did didn't understand the significance. One prominent botanist wrote back suggesting Mendel do more experiments. The paper was cited a total of 3 times in the next 35 years. 3 times. for one of the most important discoveries in the history of biology. Why was Mendel ignored? Several reasons. His mathematical approach was unusual for biology at the time. Most naturalists weren't comfortable with ratios and probability. His paper was published in an obscure journal that major scientists didn't read, and honestly, the world just wasn't ready. Darwin's theory was still being debated. The idea that heredity followed precise mathematical laws seemed too neat, too clean for the messy world of living things. Mendel became the abbot of his monastery in 1868 and spent his remaining years dealing with administrative duties and a tax dispute with the government. He died in 1884 at 61. He reportedly told a colleague near the end that his time would come. He was right, but he never got to see it. In 1900, 34 years after Mendel's paper was published, 3 botanists working independently in 3 different countries rediscovered his laws. Hugo de Vries in the Netherlands, Karl Correns in Germany, and Erich von Tschermak in Austria. Each of them was doing breeding experiments. Each of them found the same patterns Mendel had found, and each of them, when searching the literature, stumbled upon Mendel's forgotten paper. The reaction was immediate. Mendel had been right all along. His laws of inheritance were real. And suddenly, the scientific world had the missing piece that Darwin had needed. Heredity wasn't random. It wasn't blending. It followed precise rules. Mendel's factors, now called genes, were the units of inheritance. But knowing that genes exist raised an obvious next question. What are they? Physically, what is a gene? Where is it in the cell? What is it made of? Because Mendel's laws told you how traits are inherited, they didn't tell you what the actual stuff of inheritance is. That question would drive biology for the next half-century, and answering it would require a completely different set of tools and a completely different cast of characters. The search for the physical basis of heredity is one of the greatest detective stories in science. Because once scientists accepted that genes were real, the race was on to find them. And it took decades of work by dozens of brilliant people to get there. The first big clue came from looking inside cells. By the late 1800s, scientists had gotten much better at using microscopes and chemical stains to study cell interiors. They noticed these strange thread-like structures inside the nucleus that absorbed dye really well. They called them chromosomes, meaning colored bodies. And they noticed something interesting. When cells divide, the chromosomes split and distribute themselves very precisely. Each daughter cell gets exactly the same set. That kind of precision suggested chromosomes were doing something important. In 1902, 2 scientists working independently made the connection. Walter Sutton in America and Theodore Boveri in Germany both proposed that Mendel's factors, his genes, are located on chromosomes. Chromosomes come in pairs. Genes come in pairs. Chromosomes separate during cell division. Genes separate during inheritance. The parallels were too perfect to be coincidence. This was the chromosome theory of inheritance. Genes aren't floating around abstractly somewhere. They're physically sitting on chromosomes inside the nucleus of every cell. Then came Thomas Hunt Morgan. Morgan was a skeptic at first. He didn't fully buy the chromosome theory, so he set out to test it using fruit flies, tiny little Drosophila melanogaster. Fruit flies are perfect for genetics research because they breed fast, produce lots of offspring, and have only 4 pairs of chromosomes. Morgan's lab at Columbia University, which became known as the Fly Room, was legendary. Thousands of bottles full of fruit flies. The smell apparently was terrible, and a team of dedicated researchers tracking mutations across generations. And Morgan found something crucial. He discovered a white-eyed male fly among his normal red-eyed flies. When he bred this white-eyed male with red-eyed females, the pattern of inheritance was linked to sex. Only males had white eyes in certain crosses. This meant the gene for eye color was sitting on the sex chromosome. Morgan had proven that specific genes are located on specific chromosomes. He and his students went on to map dozens of genes to particular locations on fruit fly chromosomes. This was the first genetic mapping in history. Morgan won the Nobel Prize in 1933 for this work. So now scientists knew that genes live on chromosomes, but chromosomes are made of two things, protein and DNA, and the question became which one carries the genetic information. Most scientists bet on protein. Proteins are complex, varied, made of 20 different amino acids. DNA seemed too simple. It's made of only 4 building blocks, the nucleotides adenine, thymine, guanine, and cytosine. A, T, G, and C. How could something with only 4 components possibly encode all the complexity of life? Protein seemed like the obvious answer. They were wrong. In 1944, Oswald Avery and his colleagues at Rockefeller University proved that DNA, not protein, is the molecule that carries genetic information. They did this using bacteria, showing that DNA alone could transform one type of bacterium into another. The hereditary material was DNA, but many scientists still resisted. The evidence was strong, but the prejudice against DNA being too simple was deeply entrenched. It took another decade and more experiments before the scientific community fully accepted it. Meanwhile, Erwin Chargaff made a discovery that turned out to be a critical clue. He analyzed DNA from different organisms and found a consistent pattern. The amount of adenine always equaled the amount of thymine. The amount of guanine always equaled the amount of cytosine. A equals T, G equals C, always, in every organism. This was called Chargaff's rule, and it hinted at something fundamental about how DNA is structured. The bases pair up, A with TG with C, but nobody had yet figured out what the actual three-dimensional structure of DNA looked like. And this is where the story gets both thrilling and deeply unfair. In the early 1950s, there was an intense race to determine the structure of DNA. At King's College London, a physicist named Rosalind Franklin was using X-ray crystallography to study DNA fibers. This technique fires X-rays at a molecule and captures the diffraction pattern on film. From these patterns, you can deduce the molecule's shape. Franklin was meticulous, exacting, and brilliant at this work. In 1952, she produced an image known as Photo 51. It clearly showed that DNA has a helical structure. This was the key piece of evidence. At Cambridge, James Watson and Francis Crick were trying to build a physical model of DNA structure. They weren't doing experiments themselves. They were trying to piece together everyone else's data into a coherent, three-dimensional shape. And in January 1953, without Franklin's knowledge or consent, her colleague Maurice Wilkins showed Photo 51 to Watson. Watson immediately recognized what it meant. Within weeks, Watson and Crick built their famous double helix model, 2 strands of DNA wound around each other with the base pairs A with T, G with C, forming the rungs of a twisted ladder. On February 28, 1953, Crick walked into a pub in Cambridge and announced that they had found the secret of life. He wasn't exaggerating. The double helix explained everything. How genetic information is stored in the sequence of base pairs, how DNA copies itself by unzipping and using each strand as a template, how genetic information gets passed from parent to offspring. It was elegant, simple, and profound. Watson, Crick, and Wilkins shared the Nobel Prize in 1962. Franklin was not included. She had died of ovarian cancer in 1958 at just 37 years old, likely caused by her extensive exposure to X-rays during her research. Nobel Prizes are not awarded posthumously, but the deeper injustice is that her contribution was minimized for decades. Watson's memoir portrayed her dismissively. It took years before the scientific community fully acknowledged that without Franklin's data, the double helix might not have been discovered when it was. She deserved far more credit than she received in her lifetime. After the double helix, everything accelerated. Scientists cracked the genetic code, figuring out which combinations of 3 DNA bases code for which amino acids. Marshall Nierenberg and Hargobind Khorana led this effort through the 1960s. The central dogma of molecular biology was established. DNA makes RNA. RNA makes protein. Information flows in one direction. The blueprint of life had been decoded, from a monk counting peas to a twisted ladder of 4 letters. The secret of heredity, the thing Darwin couldn't explain, was finally understood. But understanding the code was just the beginning. What came next was learning how to read it, manipulate it, and eventually rewrite it. For most of human history, people had no idea why they got sick, and the explanations they came up with were honestly kind of wild. Bad air, evil spirits, God's punishment, an imbalance of bodily fluids. If you got sick in ancient Rome, your doctor might tell you that you had too much blood and the cure was to drain some out. If you got sick in medieval Europe, someone might suggest you carry flowers to ward off the poisonous air. During the Black Death, people thought the plague was spread by looking at a sick person, just making eye contact. That's how desperate and confused people were about disease. And underneath all of this confusion was a question that seems so basic to us now. Where does disease actually come from? And tied to that was an even more fundamental question that had been bugging scientists for centuries. Can life arise from nothing? This was called spontaneous generation, and for a long time, most educated people believed it was real. Leave meat out and maggots appear. Leave grain in a dark corner and mice show up. Life just emerges from non-living matter. It seemed obvious. In 1668, an Italian physician named Francesco Redi ran one of the first controlled experiments in biology. He put meat in several jars. Some were open, some were covered with gauze, some were sealed completely. Maggots appeared only in the open jars where flies could land and lay eggs. No flies, no maggots. Life wasn't appearing from nothing. It was coming from other life, but the debate didn't die. Supporters of spontaneous generation argued that maybe it doesn't work for maggots, but surely it works for microscopic organisms. Boil some broth, seal it, and microbes still appear. See? Spontaneous generation. An Italian priest named Lazzaro Spallanzani countered this in the 1760s by boiling broth longer and sealing the containers more carefully. No microbes grew, but critics said he destroyed some vital force in the air by sealing the containers too tightly. The argument went back and forth for another century. It took Louis Pasteur to finally kill spontaneous generation for good. In 1859, the same year Darwin published Origin of Species, Pasteur designed his famous swan neck flask experiment. He boiled broth in flasks with long curved necks that were open to the air but shaped so that dust and microbes would settle in the curve and never reach the broth. The broth stayed clear. No microbes grew, but if he broke the neck of the flask, allowing dust to fall in, microbes appeared within days. Air could get in. Vital force wasn't destroyed. Microbes just couldn't get past the curve. Spontaneous generation was dead, but Pasteur was just getting started. He went on to develop germ theory, the idea that specific microorganisms cause specific diseases. This sounds so obvious to us now that it's hard to appreciate how radical it was. Pasteur showed that fermentation, the process that makes wine and beer, was caused by living microorganisms, not by some chemical reaction. He showed that these microbes could be killed by heat. That's pasteurization. You see his name every time you buy milk. He developed vaccines for anthrax and rabies. The rabies vaccine in particular made him a global hero. A 9-year-old boy named Joseph Meister was bitten by a rabid dog in 1885. Without treatment, he would die. Pasteur, who wasn't even a physician, took the enormous risk of injecting the boy with his experimental vaccine. It worked. The boy survived, and Pasteur became one of the most famous scientists in the world. Working alongside Pasteur in this revolution was Robert Koch, a German physician who took germ theory and made it rigorous. Koch didn't just say microbes cause disease. He proved which specific microbe causes which specific disease. He identified the bacterium that causes anthrax. He identified the one that causes tuberculosis, the biggest killer of the 19th century. He identified the one that causes cholera, and he developed a set of criteria, now called Koch's postulates, for definitively proving that a specific organism causes a specific disease. You have to find the microbe in every case of the disease. You have to isolate it and grow it in pure culture. You have to show that it causes the disease when introduced into a healthy organism, and you have to re-isolate it from that newly sick organism. These postulates became the gold standard for identifying infectious agents, and they're still used in modified form today. The practical impact of germ theory was immediate and massive. A British surgeon named Joseph Lister started applying antiseptic techniques during surgery in the 1860s. Before Lister, surgery was basically a death sentence from infection. Surgeons operated in street clothes with unwashed hands. Operating theaters reeked of pus and rot. Post-surgical infection rates were horrifying. Lister started sterilizing instruments and wounds with carbolic acid. Infection rates plummeted. Patients stopped dying from surgery itself. Modern antiseptic and eventually aseptic surgery all traces back to Lister applying Pasteur's germ theory in the operating room. And then came the miracle drug. In 1928, a Scottish bacteriologist named Alexander Fleming came back from vacation to find that one of his Petri dishes had been contaminated by mold. Instead of throwing it away, he noticed something strange. The bacteria around the mold were dead. The mold was producing something that killed bacteria. Fleming identified the mold as Penicillium notatum and called the antibacterial substance penicillin. It took another decade and the work of Howard Florey and Ernst Boris Chain to figure out how to produce penicillin in large enough quantities to be useful. But once they did, it changed medicine forever. Infections that had been death sentences became curable overnight. During World War II, penicillin saved countless lives on the battlefield. It was hailed as a miracle drug, and it launched the antibiotic revolution. Suddenly, we had weapons against bacterial infections: streptomycin, tetracycline, erythromycin. One after another, new antibiotics were discovered and developed. But here's the thing that should keep you up at night. Bacteria evolve. They develop resistance to antibiotics, and we've been overusing antibiotics for decades. In agriculture, in medicine, sometimes for infections that don't even need them. The result is that antibiotic-resistant bacteria are spreading, superbugs that our drugs can't touch. The World Health Organization has called antibiotic resistance one of the greatest threats to global health. We might be heading toward a post-antibiotic world where a simple cut could become deadly again. The miracle that began with Fleming's moldy Petri dish might not last forever unless we find new solutions. And that's a problem biology is racing to solve right now. Now let's zoom in, way in, because everything we've talked about so far, every organism, every disease, every trait that gets inherited, it all comes down to one thing: the cell, the fundamental unit of life. And figuring that out took a surprisingly long time. Remember Robert Hooke looking at cork under his microscope in 1665 and seeing those little chambers he called cells? That was the first observation. And remember Leeuwenhoek seeing his animalcules swimming around in pond water? Those were individual cells too, though nobody understood that yet. For nearly 200 years after Hooke, scientists kept observing cells in different organisms without realizing they were looking at something universal. They saw cells in plants, they saw cells in animals, they saw single-celled organisms, but nobody put it all together into one big idea until 1838 and 1839. That's when two German scientists, Matthias Schleiden and Theodor Schwann, independently came to the same conclusion. Schleiden, a botanist, declared that all plants are made of cells. Schwann, a zoologist, declared that all animals are made of cells. Together, they established the first two principles of what we now call cell theory. All living things are composed of cells. The cell is the basic unit of life. This was huge. It meant that despite the incredible diversity of living things, from oak trees to elephants to bacteria, they all share the same fundamental building block. Life has a common architecture, but there was a missing piece. Where do cells come from? Schleiden and Schwann weren't entirely sure. Some scientists still clung to the idea that cells could arise spontaneously from non-living matter. Then, in 1855, a German physician named Rudolf Virchow made the decisive statement, Omnis cellula e cellula. All cells come from cells. No cell just appears from nothing. Every cell in your body right now came from a previous cell that divided, and that cell came from another cell that divided before it. If you trace it back far enough, every cell in every living thing connects to an unbroken chain of cell division stretching back billions of years to the very first cell that ever existed. That's a staggering thought when you really let it sink in. These three principles, all living things are made of cells, the cell is the basic unit of life, and all cells come from existing cells, form cell theory. And cell theory is one of the foundational pillars of biology alongside evolution and genetics. Everything rests on it, but knowing that cells exist and that they're important is one thing. Understanding what's actually going on inside them is another. For a long time, cells looked like little blobs of jelly under the microscope. Scientists could see the nucleus, that darker structure in the center. They could see the cell membrane on the outside, but the interior was mostly a mystery. They called it protoplasm, which is basically a fancy word for we don't know what this stuff is. That changed dramatically in the 1930s when the electron microscope was invented. Regular light microscopes can magnify things a few hundred times. Electron microscopes can magnify things hundreds of thousands of times. Suddenly, scientists could see the ultrastructure of cells, and what they found was astonishing. A cell isn't a blob of jelly, it's a factory. An incredibly complex, highly organized molecular factory. There's the nucleus, the command center, housing the DNA. There's the endoplasmic reticulum, a maze of membranes where proteins are assembled. The Golgi apparatus packages and ships those proteins to where they need to go. Lysosomes act as the recycling center, breaking down waste. The cytoskeleton provides structural support, like the scaffolding of a building. And then there are the mitochondria, the powerhouses of the cell. If you've heard one fact about cell biology, it's probably that one. Mitochondria generate the energy that keeps the cell running. Without them, complex life as we know it couldn't exist. But here's where it gets really wild. In 1967, a biologist named Lynn Margulis proposed something that most of the scientific establishment thought was absolutely crazy. She suggested that mitochondria weren't always part of our cells. They used to be free-living bacteria. Billions of years ago, a primitive cell engulfed a bacterium, but instead of digesting it, the two formed a partnership. The bacterium provided energy. The host cell provided protection and resources. Over time, this partnership became permanent. The bacterium evolved into the mitochondrion. Same thing happened with chloroplasts, the structures in plant cells that perform photosynthesis. They were once free-living photosynthetic bacteria that got swallowed up and incorporated. This is called the endosymbiotic theory, and when Margulis first proposed it, she was ridiculed. Her paper was rejected by about 15 journals before it was finally published. Scientists called it speculative nonsense, but the evidence kept piling up. Mitochondria have their own DNA, separate from the cell's nuclear DNA. That mitochondrial DNA is circular, just like bacterial DNA. Mitochondria have double membranes, exactly what you'd expect if one cell engulfed another. They replicate independently within the cell, dividing on their own schedule. They're roughly the same size as bacteria. Every single piece of evidence pointed to Margulis being right. And today, endosymbiotic theory is accepted as one of biology's great insights. The complex cells that make up every animal, plant, and fungus on this planet exist because of an ancient partnership between different types of microorganisms. We are literally built from a collaboration that happened over 1 billion years ago. Understanding cells also meant understanding how they reproduce. Cell division is how organisms grow, how wounds heal, how life continues. Mitosis is the process where a cell copies its DNA and splits into 2 identical daughter cells. Your body does this billions of times a day. Then there's meiosis, a special type of cell division that produces reproductive cells, eggs, and sperm with half the normal number of chromosomes. When an egg and sperm combine, you get a full set again. This is why you inherit half your DNA from each parent. This is the physical mechanism behind Mendel's laws. His abstract factors, his genes, are physically located on chromosomes that get divided during meiosis and recombined during fertilization. The monk's mathematics and the cell biologist's microscope were describing the same reality from different angles. And when those 2 perspectives finally came together, biology became immeasurably more powerful. So far, we've been zooming in cells, genes, DNA, the microscopic machinery of life. But now, let's zoom all the way out, because there's another way to understand living things, not by looking at what's inside them, but by looking at what's around them, how organisms interact with each other and with their environment, how everything connects to everything else. This is ecology, and its story is just as fascinating as anything happening under a microscope. The first person to really see nature this way was Alexander von Humboldt. This guy was extraordinary. Born in Prussia in 1769, Humboldt was a restless genius who wanted to understand everything about the natural world, and not in pieces, as a whole. Between 1799 and 1804, he went on an epic 5-year expedition through Central and South America, climbing volcanoes, exploring jungles, measuring everything he could: temperature, altitude, plant distribution, magnetic fields. He collected over 60,000 plant specimens. But what made Humboldt different from every other naturalist of his era was that he didn't just catalog individual species. He looked at how they connected. He noticed that similar climates produce similar vegetation even on different continents. He saw that altitude affects plant life the same way latitude does. He described nature as a web of interconnected forces. Nothing exists in isolation. Every organism is part of a larger system. Humboldt was essentially the first ecologist, though the word didn't exist yet. That word was coined by Ernst Haeckel in 1866. Haeckel, a German biologist and passionate supporter of Darwin, defined ecology as the study of the relationships between organisms and their environment. It's a simple definition, but a massive idea, because once you start thinking about relationships, things get complicated fast. Who eats whom? Who competes with whom? How does energy flow through a system? What happens when you remove one species from the chain? Charles Elton, a British ecologist working in the 1920s, started answering these questions systematically. He introduced the concept of food chains and food webs. Energy starts with plants that capture sunlight through photosynthesis. Herbivores eat the plants. Predators eat the herbivores. Decomposers break everything down when it dies and return nutrients to the soil. Energy flows in one direction. Nutrients cycle around and around. Elton also developed the idea of ecological niches. Every species has a role, a job in its ecosystem: where it lives, what it eats, when it's active, how it interacts with other species. 2 species can't occupy exactly the same niche in the same place for long. One will outcompete the other. These ideas gave ecology a framework. It wasn't just nature appreciation anymore. It was science with principles and predictions. But ecology's most powerful moment came not from a scientist in the field, but from a writer at her desk. Rachel Carson was a marine biologist and author who had already written beautifully about ocean life. But in 1962, she published Silent Spring and it changed the world. Carson documented how pesticides, particularly DDT, were devastating ecosystems. DDT was sprayed everywhere in the 1940s and 50s to kill mosquitoes and agricultural pests. It worked brilliantly for that purpose, but it didn't just kill the target insects. It accumulated in the food chain. Insects absorbed it. Birds ate the insects. The DDT concentrated in the birds' bodies, causing their eggshells to become so thin that they cracked before the chicks could develop. Bird populations were collapsing. Bald eagles, peregrine falcons, pelicans, all heading toward extinction, and nobody was connecting the dots until Carson did. The chemical industry attacked her viciously. They called her hysterical, emotional, unscientific. One executive reportedly said that because she was unmarried, she was probably a communist. The sexism was blatant and relentless, but Carson had the evidence. She had meticulously documented the science, and the public listened. Silent Spring led directly to the ban on DDT in the United States, the creation of the Environmental Protection Agency, and the birth of the modern environmental movement. One book changed the relationship between humanity and the natural world. Carson's work highlighted something ecology had been saying all along. You cannot understand an organism in isolation. Everything is connected. Spray a pesticide in a field and birds die miles away. Dump chemicals in a river and fish disappear downstream. Cut down a forest and the rainfall patterns change for entire regions. Ecology forced biology to think big, to think about systems, not just species. Edward O. Wilson pushed this thinking even further. Wilson, a Harvard biologist specializing in ants, became one of the most important voices for biodiversity in the late 20th century. He developed the theory of island biogeography with Robert MacArthur, showing that the number of species on an island is determined by a balance between immigration and extinction rates. Smaller islands farther from the mainland have fewer species. This theory applied not just to literal islands, but to any isolated habitat. A forest fragment surrounded by farmland, a lake surrounded by desert, a national park surrounded by development. Wilson warned that we were creating islands everywhere by fragmenting habitats, and on those islands, species were disappearing, and they are disappearing fast. Scientists now believe we are living through the 6th mass extinction in Earth's history. The previous 5 were caused by asteroid impacts, volcanic eruptions, and dramatic climate shifts. This one is caused by us: habitat destruction, pollution, climate change, overexploitation, invasive species. Current extinction rates are estimated to be 100 to 1,000 times higher than the natural background rate. Species are vanishing before we even discover them. Entire ecosystems are collapsing. Coral reefs are bleaching. Rainforests are shrinking. Insect populations are declining at alarming rates. The web of life that Humboldt described over 200 years ago is unraveling. And ecology is telling us that when you pull enough threads from a web, the whole thing falls apart. The question is whether we're listening. Now let's come back to evolution, because after Darwin, the story was far from over. In fact, for a few decades after Origin of Species, evolution was in trouble. Not the idea that species change over time. Most scientists accepted that by the late 1800s. The problem was the mechanism: natural selection. A lot of scientists weren't convinced it was powerful enough to drive the kind of changes Darwin described. And the reason was simple. Nobody understood how heredity actually worked. Remember, Mendel's work was sitting unread in an obscure journal. Without genetics, natural selection had a gaping hole. Critics pointed out a real problem. If traits blend together in offspring, like mixing paint, then any new advantageous variation would get diluted within a few generations. It would be swamped by breeding with normal individuals. Natural selection couldn't work if heredity was blending. Darwin knew this was a problem and never solved it. Then in 1900, Mendel was rediscovered, and ironically, this initially made things worse. Early geneticists, called Mendelians, focused on large, dramatic mutations. They thought evolution happened in big, sudden jumps, not through the gradual accumulation of small changes that Darwin described. Meanwhile, biometricians, scientists who studied variation statistically, insisted that evolution was gradual and continuous. The two camps fought bitterly for years. Mendelians said evolution is about big mutations. Biometricians said it's about small, continuous variation. Neither side would budge. It took a generation of brilliant mathematical thinkers to resolve this. In the 1920s through 1940s, Ronald Fisher, J.B.S. Haldane, and Sewall Wright independently showed that Mendelian genetics and Darwinian natural selection weren't contradictory at all. They were complementary. Mendel's discrete genes could produce the continuous variation that Darwin described. Small mutations at many genes, combined with natural selection acting on populations over time, could drive exactly the kind of gradual evolutionary change Darwin proposed. Fisher's 1930 book, The Genetical Theory of Natural Selection, was particularly influential. He translated natural selection into precise mathematical language. Evolution wasn't just a verbal argument anymore. It was equations. It was quantifiable. It was rigorous. This reconciliation became known as the modern synthesis, and it brought together genetics, natural selection, paleontology, systematics, and ecology into one unified framework. Theodosius Dobzhansky's 1937 book Genetics and the Origin of Species was the landmark text. Dobzhansky showed how genetic variation in natural populations provides the raw material for natural selection. He studied fruit fly populations in the wild and demonstrated that they contained enormous genetic diversity. Evolution wasn't just theoretical. You could see it happening in real populations. Dobzhansky also gave us one of the most famous quotes in all of biology. Nothing in biology makes sense except in the light of evolution. And he was right. Evolution is the thread that connects everything. Genetics, ecology, anatomy, behavior, medicine. Without evolution, biology is just a collection of disconnected facts. With evolution, it's a coherent story. But the modern synthesis was built before anyone knew what DNA looked like. Once molecular biology exploded in the 1950s and 60s, evolution gained an entirely new dimension. Scientists could now compare organisms not just by their anatomy or fossils, but by their DNA sequences, and the results were stunning. You share about 98.7% of your DNA with chimpanzees, about 85% with mice, about 60% with bananas. Bananas. That's how deeply connected all life on Earth is. Every living thing shares the same basic genetic code, the same DNA bases, the same system of DNA to RNA to protein. This molecular evidence confirmed what Darwin had proposed from fossils and anatomy. All life descends from a common ancestor. Molecular biology also gave us the molecular clock. The idea is that mutations accumulate in DNA at a roughly steady rate over time. By comparing how different two species' DNA sequences are, you can estimate when they diverged from their common ancestor. This revolutionized our understanding of the tree of life. We could now put dates on evolutionary events, even when the fossil record was incomplete. And then came a discovery that completely rewrote the tree of life. In 1977, Carl Woese, a microbiologist at the University of Illinois, was studying ribosomal RNA in different microorganisms. Ribosomes are the molecular machines that build proteins, and they exist in every living cell. Woese compared ribosomal RNA sequences across many species and found something nobody expected. There was a group of microorganisms that looked like bacteria under a microscope, but were genetically as different from bacteria as bacteria are from us. They were a completely separate domain of life. Woese called them archaea. Before Woese, life was divided into two categories: prokaryotes, cells without a nucleus, and eukaryotes, cells with a nucleus. After Woese, life had three domains: bacteria, archaea, and eukarya. An entire branch of the tree of life had been hiding in plain sight. Many archaea live in extreme environments, boiling hot springs, extremely salty lakes, deep-sea volcanic vents, but they're also everywhere else, in soil, in the ocean, in your gut. We just hadn't recognized them as fundamentally different from bacteria. Then things got even messier. Scientists discovered that bacteria and archaea don't just pass genes from parent to offspring vertically like we do. They also transfer genes sideways to completely unrelated organisms. This is called horizontal gene transfer. A bacterium can pick up a gene from a totally different species and incorporate it into its own genome. This means the tree of life isn't really a neat branching tree at the microbial level. It's more like a tangled web. Branches fuse together. Lineages exchange genetic material. The clean diagram in your biology textbook is an oversimplification. Reality is messier and more interesting, and modern evolutionary biology keeps revealing new surprises. Evo-Devo, evolutionary developmental biology, has shown that wildly different animals share the same toolkit of developmental genes. Hox genes control body plans in everything from fruit flies to fish to humans. A fly and a mouse use essentially the same genetic switches to determine which end is the head, where the limbs go, how segments are organized. The diversity of animal forms isn't about having different genes. It's about using the same genes differently. Small changes in when and where developmental genes are activated can produce dramatically different body plans. This deep genetic unity across the animal kingdom is one of the most beautiful discoveries in modern biology. We are all variations on the same ancient theme. In the year 2000, two rival teams stood on the brink of completing the most ambitious project in the history of biology, reading the entire human genome. Every single letter of the genetic code that builds a human being, 3 billion base pairs of DNA. The instruction manual for making you. The Human Genome Project had started in 1990 as a massive international public effort. Funded by governments, coordinated across labs in 6 countries, it was supposed to take 15 years. The approach was methodical and careful. Map the genome piece by piece, chromosome by chromosome. But then Craig Venter showed up and blew the whole thing open. Venter was a maverick scientist who thought the public project was too slow and too expensive. He founded a private company called Celera Genomics and announced he'd sequence the human genome faster and cheaper using a different technique called whole genome shotgun sequencing. Instead of carefully mapping everything first, you blast the entire genome into millions of tiny fragments, sequence them all simultaneously, and then use powerful computers to assemble the pieces back together like the world's most complicated jigsaw puzzle. The public consortium was furious. This was supposed to be a noble scientific endeavor for the benefit of all humanity, and here was this guy trying to race them for profit. The competition became intense. Both sides pushed harder, worked faster, threw more resources at the problem, and in the end, it was declared a tie. In June 2000, President Bill Clinton hosted both teams at the White House and announced that the human genome had been sequenced. The full high-quality sequence was published in 2003. Mission accomplished. And then came the surprises. Scientists had expected to find around 100,000 genes in the human genome, maybe more. After all, we're incredibly complex organisms. Surely we need a lot of genes to build something as complicated as a human being. The actual number turned out to be around 20,000. That's it. A tiny roundworm called C. elegans has about 20,000 genes too. A grape has about 30,000. We have fewer genes than a grape. This was humbling to say the least. Clearly, complexity doesn't come from simply having more genes. It comes from how those genes are regulated, when they're turned on and off, how they interact with each other, and how their products are modified after being produced. And that massive chunk of DNA that doesn't code for proteins, the roughly 98% that scientists initially dismissed as junk DNA, turns out it's not junk at all. Much of it contains regulatory sequences, switches and dimmers that control when and where genes are active. Some of it produces RNA molecules that don't make proteins, but play crucial roles in gene regulation. The genome is far more subtle and sophisticated than anyone imagined. The simple picture of genes as beads on a string, each one making one protein, was hopelessly outdated. The reality is a complex, dynamic network of interactions. This realization fed into one of the most exciting areas of modern biology: epigenetics. The word literally means above genetics. Epigenetics studies changes in gene activity that don't involve changing the DNA sequence itself. Your DNA sequence is the same in every cell of your body. A liver cell and a brain cell have identical DNA, but they behave completely differently because different genes are active in each one. Chemical tags attached to the DNA or to the proteins that DNA wraps around can silence some genes and activate others. These tags can be influenced by your environment, what you eat, what you're exposed to, how much stress you experience. Your lifestyle can literally change which genes are turned on and which are turned off. And here's where it gets really interesting. Some of these epigenetic changes can be inherited. Not changes to the DNA sequence, but changes to how the DNA is read. A parent's experiences might affect gene expression in their children, maybe even their grandchildren. Studies on the children of famine survivors have shown altered metabolism patterns that seem to be epigenetically inherited. This isn't Lamarck's inheritance of acquired characteristics exactly. Nobody's stretching their neck and passing that on. But the environment is influencing heredity in ways that go beyond simple DNA sequence. Lamarck wasn't completely right, but he wasn't completely wrong either. 2 centuries later, he's getting a partial comeback. The genomic revolution also transformed how we study the living world beyond humans. Metagenomics allows scientists to sequence all the DNA in an environmental sample, scoop up some seawater, extract the DNA, and sequence everything in it. You'll find thousands of species, most of which have never been identified or studied. This has revealed that the microbial world is vastly more diverse than anyone suspected. And some of the most important microbes live inside us. The human microbiome, the trillions of bacteria, archaea, fungi, and viruses that inhabit your body, has become one of the hottest areas of biological research. You have roughly as many microbial cells in and on your body as you have human cells. These microbes aren't just passengers. They help you digest food, train your immune system, produce vitamins, and may even influence your mood and behavior through the gut-brain axis. Disruptions to your microbiome have been linked to obesity, diabetes, depression, autoimmune diseases, and more. We're only beginning to understand how deeply intertwined our biology is with the microorganisms we carry. And then there's ancient DNA. For most of the history of genetics, studying DNA meant working with living organisms or very recently dead ones. DNA degrades over time. It was assumed that after a few thousand years, there'd be nothing left to read. But advances in sequencing technology proved that assumption wrong. Svante Pääbo, a Swedish geneticist, spent decades developing techniques to extract and sequence DNA from ancient specimens. His team sequenced the Neanderthal genome, showing that Neanderthals and modern humans interbred. If your ancestors came from outside of Africa, you likely carry between 1 and 4% Neanderthal DNA right now. They also discovered an entirely new group of ancient humans called the Denisovans, known only from a tiny finger bone and a few teeth found in a Siberian cave. Ancient DNA has revolutionized our understanding of human evolution and migration. Pääbo won the Nobel Prize in 2022 for this work. We can now read the genomes of organisms that died tens of thousands of years ago. The past is no longer silent. It has a genetic voice, and we've learned to listen. L-Immune System into a precision gene editing tool. It's called CRISPR-Cas9, and to understand why it's so revolutionary, you need to know where it came from. Bacteria have been fighting viruses for billions of years. When a bacterium survives a viral attack, it stores a snippet of the virus's DNA in its own genome, in a region called CRISPR, clustered regularly interspaced short palindromic repeats. That's a mouthful, which is why everyone just says CRISPR. These stored snippets act like a molecular mugshot file. If the same virus attacks again, the bacterium uses that stored sequence to recognize the invader and deploys a protein called Cas9 to cut the viral DNA apart. It's an immune system, a molecular search and destroy mechanism that bacteria have been using for billions of years. What Doudna and Charpentier realized was that you could reprogram this system. You could design a guide RNA that matches any DNA sequence you want, not just viral DNA. Attach that guide to the Cas9 protein, introduce it into any cell, and it will find the matching sequence and cut it. Right there, precisely. You can knock out a gene by cutting it. You can delete a section. You can even insert new DNA at the cut site. It's like a molecular word processor. Find, cut, paste, replace. Except instead of editing a document, you're editing the blueprint of a living organism. The scientific world went crazy. CRISPR was faster, cheaper, and more accurate than any previous gene editing technology. Previous tools existed, but they were clunky, expensive, and hard to use. CRISPR democratized gene editing. Suddenly, any competent molecular biology lab in the world could edit genes. The cost dropped from thousands of dollars to under $100. Experiments that used to take months could be done in weeks. Doudna and Charpentier won the Nobel Prize in Chemistry in 2020. Two women sharing a Nobel for one of the most consequential discoveries of the 21st century. And the applications are already here. In December 2023, the first CRISPR-based therapy was approved for clinical use. It treats sickle cell disease, a painful and debilitating genetic condition caused by a single mutation in one gene. Doctors take a patient's blood stem cells, edit the faulty gene using CRISPR, and put the corrected cells back. The results have been remarkable. Patients who suffered agonizing pain crises their entire lives are now symptom-free. One mutation, one edit, one cure. In agriculture, CRISPR is being used to develop disease-resistant crops, drought-tolerant varieties, and more nutritious foods. Scientists have created mushrooms that don't brown, tomatoes with higher nutritional content, and rice varieties that can withstand flooding. Unlike traditional genetic modification, which inserts foreign DNA from other species, CRISPR can make precise changes within an organism's own genome. It's faster and more targeted than anything we've had before. But with this power comes a question that keeps scientists up at night. Where's the line? In November 2018, a Chinese scientist named He Jiankui announced that he had used CRISPR to edit human embryos, not just any embryos, embryos that were implanted and carried to term. Twin girls were born with edited genes. He claimed he had disabled a gene called CCR5 to make the girls resistant to HIV. The scientific community erupted. This was not a celebrated breakthrough. It was a scandal. He Jiankui had crossed a line that the global scientific community had explicitly agreed should not be crossed. Not yet. Not like this. The edits were made without proper oversight, without adequate safety testing, without understanding the potential long-term consequences. Editing an embryo means the changes get passed to every cell in the body and to all future generations. You're not just editing one person, you're editing their children and their children's children. The risks of unintended effects are enormous, and we don't understand them well enough yet. He Jiankui was condemned by scientists worldwide, fired from his university, and sentenced to 3 years in prison by the Chinese government. But the damage was done. The genie was out of the bottle. He had proven it was technically possible. And once something is proven possible, it becomes much harder to prevent others from trying it. Meanwhile, biology is pushing even further. Craig Venter, the same maverick who raced the Public Genome Project, announced in 2010 that his team had created the first synthetic organism. They designed an entire bacterial genome on a computer, built it chemically from scratch in the lab, and inserted it into a cell whose own DNA had been removed. The cell booted up the synthetic genome and started living, dividing, growing, a living organism running on software written by humans. This is synthetic biology, designing life from the ground up. Scientists are now engineering microorganisms to produce biofuels, manufacture drugs, clean up pollution, and produce materials that currently require petroleum. We're not just editing the book of life. We're writing new chapters from scratch. And then there are gene drives. This might be the most powerful and most terrifying application of CRISPR. Normally, when an organism reproduces, each gene has a 50-50 chance of being passed to offspring. A gene drive hacks that system. It ensures that a particular engineered gene gets inherited by nearly all offspring, not just half. This means you could release a handful of engineered organisms into a wild population, and within a few generations, the engineered gene would spread through the entire species. The most discussed application is eliminating malaria by engineering mosquitoes that can't carry the malaria parasite. Malaria kills over 600,000 people every year, mostly children in sub-Saharan Africa. A gene drive could potentially wipe out the disease by making mosquitoes unable to transmit it. That sounds miraculous, but think about what it means. We'd be making a permanent genetic change to a wild species across entire continents. What if there are unintended ecological consequences? What if eliminating one species of mosquito collapses a food web? What if the gene drive mutates and does something we didn't predict? Who gets to make this decision? One country? One scientist? The whole world? This is where biology stands right now. We have the power to read the code of life, to edit it with precision, to write it from scratch, and to spread those edits through wild populations. We can cure genetic diseases. We can engineer new organisms. We can potentially eliminate ancient plagues. But every one of these powers comes with risks that we don't fully understand and ethical questions that we haven't fully answered. For the first time in the 4 billion-year history of life on Earth, one species has the ability to deliberately rewrite the rules. The question isn't whether we can. We already can. The question is whether we're wise enough to know when we should and when we shouldn't. And honestly, the history of biology doesn't always inspire confidence on that front, but it does show us something else, that when humans pay attention, when they observe carefully, think rigorously, and hold each other accountable, they can figure out extraordinary things. The challenge now is making sure the figuring out stays ahead of the consequences. So let's step back for a moment, because when you look at the entire arc of biology, from those first Egyptian physicians pulling organs out of mummies to scientists today rewriting the genetic code of living embryos, a pattern emerges, and it's the same pattern you see in every great scientific story. It starts with observation. People looking at the world around them and asking, what is that? Aristotle classifying animals, Vesalius cutting open bodies, Hooke peering through his microscope, just looking, carefully, patiently, looking. Then comes the search for general principles. Darwin seeing natural selection behind the diversity of life. Mendel finding mathematical ratios behind inheritance. Schleiden and Schwann realizing that every living thing shares the same cellular building blocks. Individual observations start connecting into bigger ideas. Then come abstract frameworks, the modern synthesis unifying genetics and evolution, molecular biology decoding the central dogma, ecology mapping the web of relationships that sustain life, big theories that explain not just individual facts, but entire categories of phenomena. And finally, revolutionary applications, vaccines, antibiotics, gene therapy, CRISPR. The understanding becomes power, the knowledge becomes technology, and the world changes. This arc, observation to principles, to frameworks, to applications, has repeated itself over and over across the history of biology. And each time, people thought they'd reached the end. Each time, they thought they finally understood life. And each time, a new tool or a new idea revealed another layer that nobody suspected was there. The microscope revealed cells. Nobody knew life had building blocks that small. X-ray crystallography revealed the double helix. Nobody knew heredity had a physical structure that elegant. DNA sequencing revealed the genome. Nobody expected junk DNA, epigenetics, or the staggering fact that we have fewer genes than a grape. CRISPR gave us the ability to edit that genome. Nobody imagined we'd go from reading the code to rewriting it within a single generation. And here's what's beautiful about biology's story. The great themes aren't separate. They're all connected. Evolution explains why organisms are the way they are. Genetics explains how traits are passed down and how variation arises. Cell biology explains the machinery that makes it all work. Ecology explains how organisms fit together into systems. Pull on any one thread and you find it connected to all the others. You can't understand evolution without genetics. You can't understand genetics without cell biology. You can't understand ecology without evolution. It's one story told from different angles. Dobzhansky was right. Nothing in biology makes sense except in the light of evolution. But you could also say nothing in biology makes sense except in the light of cells, or genetics, or ecology. They're all load-bearing walls. Remove anyone and the whole structure wobbles, and we're nowhere near finished. The biggest questions in biology are still unanswered. How did life begin? We know that life has existed on Earth for at least 3.5 billion years. We know that all life shares a common ancestor. But we don't know how that first living thing arose from non-living chemistry. How did molecules become cells? How did chemistry become biology? Scientists have theories: RNA world hypothesis, hydrothermal vent origins, primordial soup scenarios. But nobody has definitively answered the question. The origin of life remains one of the deepest unsolved problems in all of science. Then there's consciousness. How does a 3-pound lump of neurons inside your skull produce the experience of being you? How do electrical and chemical signals become thoughts, feelings, awareness? We can map the brain. We can identify which regions light up during different activities, but we don't understand how physical processes give rise to subjective experience. This is sometimes called the hard problem of consciousness, and it might be the hardest question biology has ever faced. We still haven't mapped the full tree of life. Scientists estimate there are somewhere between 8 and 10 million species on Earth. We've identified and described maybe 1.5 million of them. The vast majority of life on this planet is still unknown to science. Most of it is microbial, entire ecosystems in deep ocean sediments, deep underground, in extreme environments that we've barely begun to explore. And then there's the question that might be the biggest of all. Is there life beyond Earth? Are we alone in the universe? With billions of galaxies, each containing billions of stars, each potentially hosting planets, the odds seem overwhelming that life exists elsewhere, but we haven't found it yet. And if we do find it, everything we think we know about biology might need to be reconsidered. Is DNA universal? Is cell structure universal? Or did life elsewhere find completely different solutions? From a monk carefully counting pea plants in a monastery garden to scientists editing the DNA of human embryos in a laboratory. From a Dutch merchant squinting through a handmade lens at pond water to researchers sequencing the genome of a Neanderthal who died 50,000 years ago. Biology has come impossibly, breathtakingly far, and we're still just getting started. So here we are, at the end of a story that spans thousands of years, dozens of civilizations, and some of the most brilliant and stubborn and courageous minds who ever lived. From Egyptian embalmers pulling organs out of bodies and accidentally learning anatomy, from Aristotle getting his hands dirty dissecting animals in ancient Greece, from Sushruta performing surgeries in India that wouldn't be matched in Europe for centuries, from Islamic scholars preserving and advancing knowledge while Europe slept, from Vesalius finally picking up the scalpel and proving that the old authorities were wrong, from Leeuwenhoek discovering an invisible universe in a drop of pond water, from Linnaeus giving every living thing a name, from Darwin sitting on the most dangerous idea in history for 20 years, because he was terrified of what it meant. From Mendel counting peas in silence, knowing he'd found something extraordinary, dying before anyone believed him. From Franklin producing the image that revealed the structure of life and never getting the credit she deserved. From Margulis being told she was crazy for suggesting our cells are built from ancient partnerships. From Carson being called hysterical for telling the truth about what we were doing to the natural world. From Doudna and Charpentier handing humanity the power to rewrite the code of life itself. Every single one of these people did the same thing. They looked at the living world and refused to accept that the current explanation was good enough. They pushed further. They questioned authority. They observed more carefully. They followed the evidence even when it led somewhere uncomfortable. And that's what biology really is. Not a subject you memorize for an exam. Not a list of terms and diagrams and Latin names. It's a way of looking at the world. A 4,000-year-old tradition of asking what is life, how does it work, and why does it do what it does? And every generation gets a little closer to the answer. And every generation discovers that the answer is more complex, more beautiful, and more strange than anyone imagined. The next chapter of this story is being written right now in labs and forests and oceans and hospitals all over the world. And we have no idea what they'll discover next. But if history is any guide, it'll be something nobody expected, something that makes us rethink what we thought we knew, something that reveals yet another layer beneath the layers we've already uncovered, because that's what biology does. It keeps surprising us, and it's nowhere close to being done.